So let's start with a little background information. By the 1960s, it was known that DNA was used to synthesize RNA, and past experiments were implying that RNA is used as a template for protein synthesis. In 1961, Marshall Nirenberg and Heinrich Matthaei conducted an experiment to try and prove that RNA is in fact used for protein synthesis, where specific sequences of RNA code for a specific amino acid. Here's a quick recap of their procedure. First, the two scientists prepared extracts from E. coli cells that consisted of every part required for protein synthesis but with no mRNA. They ran this extract and discovered that without RNA, no protein synthesis occurred. So they added RNA to the mix, and lo and behold, the extract produced proteins. This RNA was a specific synthetic RNA with only uridine nucleotides known as poly-U. The experiment revealed that the poly-U RNA made a protein consisting entirely of phenylalanine amino acids. Similar experiments were conducted that found that poly-A, all adenine nucleotides, created an all lysine protein, and poly-C, all cytosine nucleotides, created an all proline protein. As a result, Nirenberg and Matthaei discovered that RNA was required for protein synthesis, and the specific RNA sequence determined what proteins were formed. This experiment inspired more questions. How many nucleotides were required to code for these amino acids? Or in other words, how many nucleotides are in a codon? These questions were answered in the experiment conducted by Marshall Nirenberg and Philip Lader. The goal of their experiment was to determine the length of codons using a triple binding assay. Two different assay mixtures were created, run, and washed through a special filter. One mixture contained ribosomes and tRNA that was charged with radioactive phenylalanine. The other mixture contained these plus poly-U RNA. These two assays were poured through a special filter that the ribosomes stuck to as the rest of the mixture fell through after a wash. After being poured and washed through the filters, only one was radioactive. The scientists observed that the mixture with the poly-U RNA made a positive radioactive reaction, while the mixture without poly-U had no reaction. Nirenberg and later discovered that using 3 or more uracil nucleotides showed radioactivity, whereas using only 2 showed no radioactivity. They realized that the minimum number of nucleotides that produced reactivity was the number of nucleotides in a codon, 3. From these experiments, they deciphered the genetic code and identified 61 codons that translate into amino acids. This went on to create the DNA codon table that we still use today.